Hi friends, today we are going to discuss glycolysis process, the second phase, Payoff phase. In this process, there will be a 5 different phase with 5 different coenzymes are involved for this process. So, in this process, we are going to with the chemical reaction, we are going to receive 4 ATP molecule at the end of the pyruvate process, pyruvate. So glucose is converted into pyruvate. We can call it as a glycolysis. So whatever the foodstuff we consume during eating, either carbohydrate, protein, or fat, this will be converted into glucose with the help of liver. So if liver is not involved in this process, definitely there will be no glucose availability in the blood. So once insulin is released from pancreatic secretion, then only your glucose may get entry into the cellular activity, that is called glycolysis. So without insulin, it is not possible of converting your glucose into energy molecule for your working muscle. So for this process, the total metabolic process is 21 stage. The glycolysis is 11 different coenzymes, and then Krebs cycle again 8 stages, 8 coenzymes, and then finally electron transfer chain. It will be involved three stages. So totally 21 stages, 22 coenzymes are involved to utilize glucose as a energy fuel for your working muscle and maintain your a constant and normal body function during your day-to-day activities. Let us discuss step by step in the payoff phase. In the payoff phase, we are going to receive or releasing 4 ATP molecule by investing 2 ATP in the investing phase. For every business, we have to spend something and process it, and finally you will get some return. Same like in the investment phase, we have to spend 2 ATP molecule to initiate the metabolic process, glycolytic process. So at the second phase, payoff phase, during this payoff phase, same like you will get some energy sources for your working muscle. This glycolytic process is carried out within the cell cytosol or above mitochondrial reaction. So let us now we are going to discuss start with sixth stage, that is oxidation of glyceraldehyde 3-phosphate into 1,3-biphosphoglycerate. Here two processes are carried out. One is removal of hydrogen from its chemical bond, and the another one is again we are going to inclusion of one more phosphate into the chemical bond. So make your chemical bond more powerful and energy yielding nature. So if we include more thing into the bucket, then you will get a bunch of energy sources. For this process, we need glyceraldehyde 3-phosphate dehydrogenase. So as I told, oxidation and dehydrogenase is carried out in this sixth stage. The process is very simple. The glyceraldehyde 3-phosphate is converted into 1,3-biphosphoglycerate with the help of glyceraldehyde 3-phosphate dehydrogenase. So, in this process, first step, NAD is converted into NADH by releasing by removal of one hydrogen from glyceraldehyde 3-phosphate. This is very important conversion in step 1. In step 2, oxidative phosphorylated process, and by this process we are going to addition one more phosphate into glyceraldehyde 3 phosphate and make it as a 1,3 biphosphoglycerate. Since two molecules of glyceraldehyde are formed from 1,3 biphosphoglycerate. So in this process, the another one is the two NADH are generated in this phase. The process is simple. To process oxidation or inclusion of one phosphate into glyceraldehyde 3 phosphate and make it as a 1,3 biphosphoglycerate. And in the glyceraldehyde 3 phosphate, we are going to remove one phosphate hydrogen from glyceraldehyde 3 phosphate and make it as a 1,3 biphosphoglycerate. Let us discuss with chemical bond. So here in this process we have to include one inorganic phosphate, 2 NADH. So in this inclusion of one phosphate, here in this chemical bond, glyceraldehyde 3 phosphate, it consists of only one phosphate. By inclusion of one inorganic phosphate, we can add one more phosphate in this chemical bond, so that the chemical bond is more powerful to yield more ATP in the further step. So the NAD is converted into NADH and one hydrogen is released. That is called dehydrogenase process. So oxidation process, inclusion of one phosphate in the chemical bond. So the process, this is first payoff phase already I, 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 we have discussed. The glyceraldehyde 3 phosphate is oxidized by coenzyme nicotinamide adenine dinucleate is converted into NADH by releasing of one hydrogen from the glyceraldehyde 3 phosphate. And seventh stage is very simple. The phosphoryl group is transferred from 1,3 biphosphoglycerate. So we can say simply the 1,3 biphosphate glycerate is releasing one phosphate group, and those phosphate group is added to adenosine diphosphate and make it as a adenosine triphosphate. So that for this process, the phosphoglycerate kinase are involved to make the function more effectively. In the seventh step, it is going to release 2 ATP return. So this is the first step where you can return back your ATP into 2 ATP. Actually, in the energy investment phase, we have spent 2, but here we get it back. Sorry, 1 ATP is involved in energy investing phase. Here you will return back as a 2 ATP molecule. The process is it involves transfer of one phosphate group from 1,3 biphosphate glycerate. So this phosphate is combined with adenosine diphosphate and make it as a adenosine triphosphate. In this process, since two molecules of 1,3 biphosphoglycerate are formed from 1 mole of glucose and 2 ATPs are generated in this phase, so you'll get 2 ATP for your working muscle. So these ATP can be utilized as an energy sources for your working muscle. So in the seventh stage, we can discuss with chemical bond. So sixth stage, we have added one phosphate in this base. In the seventh stage, we are going to remove it with help of phosphoglycerate kinase or phosphoryl transfer from 1,3 biphosphoglycerate to 3 phosphoglycerate. This one phosphate is combined with adenosine diphosphate and make it as a adenosine triphosphate. This is the process. In this step, 1,3-biphosphoglycerate is converted into 3-phosphoglycerate with the help of phosphoglycerate kinase enzyme, and phosphoryl is released from 1,3-biphosphate, and those 1-phosphate is combined with adenosine diphosphate and make it as a adenosine triphosphate. Again, remember in this step, 1 mole of glucose is released to ATP molecule, and we are going to discuss 8th step, the conversion of 3-phosphoglycerate into 2-phosphoglycerate. For this process, we needed phosphoglycerate mutase. Okay, this phosphoglycerate mutase, it will change over the base 3 to base 2. So this is the entire process of step 8. The process is, the phosphoryl group is 3-phosphoglycerate is shifted from C3 position to C2 position in the chemical bond, which yield or which convert 3-phosphoglycerate into 2-phosphoglycerate. For this process, we need magnesium. This magnesium has to combine with phosphoglycerate mutase. Then only this process can be switch over from phase 3 to phase 2. So the process will be more active. So we are going to discuss with chemical bond. So this is base 1, base 2, base 3. So in this base 3, we have the 1 phosphate. This phosphate is shifted to base 2. That is the main process. For this process, magnesium are involved. So in this step, phosphoryl group 3-phosphoglycerate is shifted to C2 position. That is the main process, which yield phosphoglycerate, 2-phosphoglycerates, and the reaction is catalyzed the enzyme phosphoglycerate mutase, which require magnesium ion for its activity. So 8th position, the 8th phase, there will be no releasing of ATP. So 9th position, dehydration of 2-phosphoglycerate to make it as a phosphoenol pyruvate. For this process, we are going to remove one more hydrogen ion from the chemical bond with help of enolase enzyme. The process is 2-phosphoglycerate is converted into phosphoenol pyruvate. Finally, the pyruvate will be separated in the 10th stage. Now we are going to remove one hydrogen from 2-phosphoglycerate by activating the process by inclusion of enzyme enolase coenzyme. So in the chemical bond, OH is at 3rd position and hydrogen at 2nd position. This hydrogen and OH, here one more hydrogen, so H2O. That means the removed, it will release as a water or removed H2O from its chemical bond and make your system or chemical bond nature phosphoenol pyruvate. So reaction is catalyzed by enzyme enolase from phosphoenol pyruvate. So with chemical reaction, we can see here, as I already mentioned, in the third base and second base, in second base one hydrogen, in third base one OH. This will combine together and make it as a water by dehydration process. So this is the only change in this position with help of enolase. So phosphoenol pyruvate is produced 2-phosphoglycerate. It is because of releasing water molecule from its chemical bond, and this reaction is catalyzed enzyme enolase phosphoenol pyruvate. And the 10th stage, this is last phase of Payoff phase, the transfer of phosphoryl group. So in the ninth stage, there will be only one phosphate. Those phosphate can be detached from its chemical bond with the help of pyruvate kinase. The process is phosphoryl group is released from phosphoenol pyruvate. The phosphate is combined with adenosine diphosphate and make the or joined together adenosine diphosphate on one phosphate and leads to production of adenosine triphosphate in this phase. So this is second energy yielding phase where the adenosine diphosphate combined with the one phosphate and make it as a ATP. ATP is released in this stage, so we can call it as a energy yielding phase in the 10th. The phosphoenol pyruvate is converted into enol and then form a pyruvate. This pyruvate is very essential to switch over the glycolytic process into Krebs cycle process. So without pyruvate with the 3-carbon molecule, it is not possible of processing or further proceeding of uh, explorate of energy sources into multiply. So in this way, finally glucose is converted into pyruvate with the help of aerobic condition. Though and then this pyruvate enter into citric acid cycle by converting into acetyl CoA. So citric acid cycle, it will be released in the 10th stage, but we have to again convert pyruvate into acetyl CoA. Then only it will enter into Krebs cycle chemical reaction with the help of oxygen. So let us see the changes in chemical bond. So finally, in the 9th stage, there will be only one phosphate. This phosphate is combined with adenosine diphosphate and make it as a ATP. So your phosphoenol pyruvate is converted into pyruvate. So the remaining chemical bond is very simple. 1, 2, 3, 3-carbon molecule. We can call it as a pyruvate. So this is the second energy releasing phase of glycolytic process, and where phosphoryl group is released from phosphoenol pyruvate, and those one phosphate is combined with adenosine diphosphate, and it start to production of 2 ATP adenosine triphosphate in the chemical bond. So this reaction yield 2 molecule of pyruvate, 2 molecule of ATP. So 2 pyruvate with one carbon with the 3 carbon with the glucose, 3 carbon with the one more glucose it will be. So this pyruvate, it will converted into acetyl CoA, enter into the Krebs cycle process. This is about glycolytic process, start from first stage to 10th stage. From first stage, glucose is converted and make it as a glyceraldehyde 3 phosphate in the 5th stage. And this is called energy investment phase. And from 6th stage, glyceraldehyde 3 phosphate can be further convert and releasing of 4 energy molecule 4 ATP and make it as a pyruvate, and then finally it will enter as a acetyl CoA in the Krebs cycle production. So in the Krebs cycle production only energy can be multiplied into 36 energy molecule at the end of Krebs cycle. So on the whole, the Payoff phase in the 6th position, 3 glyceraldehyde 3 the phosphoglyceride 3 phosphoglyceride, it will be added one more phosphate and make it as a 1,3 diphosphoglycerate. Okay, so this is sixth phase. We are adding one phosphate and make the energy chemical bond more energy one. And seventh phase, already added one phosphate can be released from its chemical bond and make it as a ATP. One phosphate is combined with adenosine diphosphate, make it as a adenosine triphosphate. So in the seventh ending phase, 3 carbon 1 phosphate, that is called 3 phosphoglyceric acid. So in the eighth stage, the phosphate group is transferred from C3 position to C2 position. So that makes the 2 phosphoglyceric acid. From its chemical bond, again it is releasing water molecule. So it make it as a phosphoenol pyruvate by releasing one more phosphate and the last phosphate from its chemical bond. So those phosphate it will combine with ADP and make it as a ATP. Finally, there will be a 3 carbon molecule with glucose. So it will be enter as a acetyl CoA into the mitochondrial reaction, that is called the Krebs cycle. In the Krebs cycle, we are going to discuss in a separate video. So up to this, the Payoff phase is ended. So on the whole process, there will be a 10 stages. Each 5 stages we have divided into energy investment phase, energy Payoff phase. Whatever the food you consume, it will be converted as a glucose with the help of liver. So glucose can be converted glucose 6 phosphate in first stage, and then the second stage fructose 6 phosphate is derived with help of PGI, and third stage one more phosphate is included and fructose 1 6 biphosphate is generated, and fourth stages it will be divided into two, that is called a dihydroxyoctone phosphate. This will be inactive. It will support to glyceraldehyde 3 phosphate and make the cyclic process continue. Okay, up to this is called the energy investment phase, and it will be enter the second phase is Payoff phase, that is called 1,3 biphosphate phosphoglycerate, and then with the PGK, 2 ATP is released from its chemical bond and make it as a 3 phosphoglycerate. This is seventh stage, and then 3 phosphoglycerate is converted into C3 position to C2 and make it as a 2 phosphoglycerate. With the enolase support, it will be again converted as a phosphoenol pyruvate. So the enolase is again it is releasing from its chemical bond two more ATP from its chemical bond and make it as a pyruvate. This pyruvate is going to enter into Krebs cycle, that is separate process. We'll see next video the Krebs cycle process. So at the end, this is up to this glycolytic process where 2 ATPs invest in this phase and 4 ATPs regenerated. So 4 ATPs released from its chemical bond. So the glucose can be converted as a pyruvate, and the pyruvate converted again and make it as a acetyl CoA, finally enter into Krebs cycle. So totally you'll get at the end of all this cyclic process, glycolytic and Krebs cycle process, you'll get 36 energy molecule, actually 38, but we have to deletion of 2 ATP which was invested in the beginning phase. So 36 net ATPs released from Krebs cycle. This is the entire process of glycolytic and Krebs cycle. Because of this cycle, you will get lot of energy during your day-to activities to maintain proper physiological and physical performance during any physical activity or day-to activities. So if you can understand this process, definitely it will be a very useful for your career, and you will be a more energetic and strong personality in this society. So in order to make you more powerful person in this world, we have to consider all those things. First thing, right kind of food intake. This food intake is converted as a glucose with help of liver. Liver is more important than any other organ in this in our body. So glucose, again we need one more hormone that is called insulin from pancreatic secretion. Without insulin, glucose may not be enter into the cytosol or cellular activity. So above mitochondrial, there will be a 10 reaction, and after generating a pyruvate acid, it will be converted as a acetyl CoA, and Krebs cycle there will be 8. So totally 18 plus in the electron transport chain again 3. So totally 21 coenzymes, and 21 uh, steps are involved to make you more energetic in this world. Thank you.